Hi friends, welcome to biologyexamsforyou.com. In this video we will be discussing 50 important facts on amino acids, a quick revision that will help you for your exams. Point number 1. Amino acids are building blocks of proteins. Point number 2. Amino acids are joined by peptide bond. Point number 3. Naturally occurring amino acids are levorotatory except glycine. Glycine is asymmetrical. So let us see what is an amino acid. Amino acid is having a central carbon atom to which an amino group is attached. On the other side there is a carboxyl group COOH. Then there is a hydrogen and this is a side chain. Side chain, if it is hydrogen, which is the simplest amino acid, that is glycine. Two amino acids are joined by peptide bond. This C double bond O NH bond is called as a peptide bond, which is formed by dehydration condensation reaction, that is by the removal of water molecule. We have given a detailed video on how peptide bond is formed. You can refer that for more. Point number 4. D amino acids are rarely found in bacterial cell walls and certain antibiotics like polymyxin, actinomycin D, etc. Point number 5. Amino acids can exist as ampholytes or sweeter ions. That means amino acid has both positive and negative charge in aqueous solution. As you can see, this amino group becomes NH3 plus and this carboxyl group becomes COO minus. Then we call it as sweeter ions. Moving into amino acid classification. Point number 6. Essential amino acids are amino acids that should be obtained through diet. 9 in numbers, it can be remembered easily as PVT Timhal. Phenylalanine, valine, tryptophan, threonine, isoleucine, methionine, histidine, leucine and lysine. Point number 7. Non-essential amino acids which are synthesized in the body. Point number 8. There are conditional amino acids or semi-essential amino acids that are required at certain specific condition, maybe during stress or maybe some other physiological conditions. Include arginine, cysteine, glutamine, tyrosine, glycine, etc. Some amino acids are produced at older age but not synthesized in infants. Such amino acids that is specifically needed at certain conditions are called as semi-essential amino acids. Point number 9. There are 20 standard amino acids or most common amino acids that are incorporated into proteins. Point number 10. 21st amino acid identified is selenocysteine and 22nd is pyrrolysine, that is not in humans but present in archaeobacteria. Point number 11. Glycine is the simplest amino acid which is achiral, means it is asymmetrical amino acid. And this is glycine. As you can see, side group is hydrogen, then it is glycine. Point number 12. Glycine is a major precursor in the biosynthesis of porphyrin nucleus in hemoglobin, cytochromes and chlorophyll. Point number 13. Glycine is an inhibitory neurotransmitter. Point number 14. Collagen, which is the most abundant structural protein in the body, contains high amount of glycine. Every third or fourth amino acid in collagen is glycine. Point number 15. Proline and glycine are known as helix breakers. Point number 16. Serine is a precursor of glycine biosynthesis. Serine is an amino acid that is involved in the synthesis of many other amino acids. 17. The two sulfur-containing amino acids are cysteine and methionine. Whereas cysteine can form disulfide bonds. And this is cysteine. As you can see, there is sulfhydryl group CH2SH group, which is a sulfhydryl group. Point number 19. Cysteine forms disulfide bond between two cysteines forming cysteine. Point number 19. Methionine cannot form disulfide bond as methionine doesn't have SH group, it is having SCH3 group. Point number 20. Start codon AUG codes for methionine in eukaryotes and archaea, whereas formyl methionine is a start codon in prokaryotes. Point number 21. Methionine is a precursor of ethylene, the gaseous plant hormone that is involved in ripening. Point number 22. Protein disulfide isomerase or PDI is an enzyme that catalyzes the formation of disulfide bonds. And disulfide bond formation occurs in the highly oxidizing environment of ER during post-translational modification of protein. Point number 23. Methionine. It provides methyl group in many reactions as S-adenosyl methionine or SAM. Point number 23. Histidine is having an imidazole side chain which has a pK of 6.8. pH of cytoplasm is having the buffering capacity. Point number 24. Histidine is a precursor of histamine which is involved in inflammatory reactions. Point number 25. Histidine is a precursor of antioxidant ergothioneine in Neurospora crassa, which is the fungus that is widely used in research in tetrad analysis and also called as the Drosophila of the fungal world. Point number 26. Tryptophan is an aromatic amino acid and also the bulkiest amino acid. Tryptophan is a precursor to neurotransmitter serotonin, then hormone melatonin and vitamin B3. Point number 27. It is also the precursor of plant hormone auxins like IAA and nicotinamide. Point number 28. Plants and microorganisms synthesize tryptophan through shikimic pathway. Point number 29. Intrinsic fluorescence of protein is primarily due to the excitation of tryptophan residues. Point number 30. Tyrosine. It forms skin pigment melanin and two other hormones, thyroxine and adrenaline. Point number 31. In photosynthesis, in photosystem 2, tyrosine acts as an electron donor in the reduction of oxidized chlorophyll. Point number 32. Tyrosine phosphorylation is a key step in signal transduction process in the regulation of enzymatic activity. We call the protein as tyrosine kinase. Point number 33. There is a hypothesis called alanine world hypothesis that proposes that one of the earliest amino acid that is to be included in the ribosome mediated biosynthesis of proteins is alanine. Point number 34. Alanine scanning is a site-directed mutagenesis technique to determine the role of specific protein amino acid residue to the stability or function of a given protein. Point number 35. The amino acid beta-alanine forms coenzyme A and vitamin pantothenic acid. Point number 36. Heterocyclic amino acids are tryptophan and histidine. Tryptophan is the bulkiest amino acid which is having an indole ring. Acidic amino acids include glutamic acid and aspartic acid. 37. Glutamic acid is the most common amino acid that undergoes oxidative deamination. Basic amino acids include lysine and arginine. Arginine contain guanidium group and histones are rich in basic amino acid lysine and arginine, as histones are positively charged whereas DNA is negatively charged, that is wrapped around positively charged histones. Point number 38. Neutral amino acids include glycine, valine and phenylalanine. Point number 39. Alcoholic amino acid or amino acids with hydroxyl group include serine and threonine. Point number 40. Aromatic amino acids include tryptophan, tyrosine and phenylalanine. All are having strong absorbance of light, and absorbance of light by proteins at 280 nanometer is due to the presence of these aromatic amino acids. Point number 41. Sulfur containing amino acids are cysteine and methionine. Point number 42. Amino acids with amide group are asparagine and glutamine. Point number 43. Proline is a secondary amino acid. Point number 44. Selenocysteine is the 21st amino acid present in human protein. Point number 45. Uncommon amino acids include collagen, the most abundant structural protein. There is 4-hydroxyproline and hydroxyglycine. In myosin, that is muscle protein, there is methyl-lysine. And also in prothrombin, blood clotting protein, there is carboxyglutamate acid. All are uncommon amino acids. Point number 46. Non-protein amino acids include ornithine and citrulline, that is involved in the synthesis of arginine and in urea cycle. Point number 47. Glutathione is an antioxidant which consists of glutamic acid, glycine and cysteine. Point number 48. Aspartame, an artificial sweetener also known as NutraSweet. It's a synthetic dipeptide which is made up of aspartic acid and phenylalanine, chemically called as L-aspartyl L-phenylalanine. Point number 49. Neurotransmitters include gamma amino butyric acid, a derivative of glutamic acid. Then there is dopamine, derivative of tyrosine. Then there is glycine, which is an inhibitory neurotransmitter. Point number 50. Sickle cell hemoglobin differs from normal hemoglobin by a single amino acid. In the beta chain of sickle cell hemoglobin, glutamic acid, which is a negatively charged amino acid, is replaced by valine, a hydrophobic amino acid that causes the sickle shape. Hope you are benefited from this video. Thank you so much for your support. If you like this video, please subscribe, support and share. If you are having some more points to add, please drop that points in the comments so that others are also benefited. Thank you so much.